I was asked at short notice to talk to you about this topical issue, which I'm sure is something that interests everyone at the moment. So this will be a brief presentation just highlighting what has happened up to now, how we discovered this variant, and what we need to bear in mind about it. It's obviously a rapidly developing situation, and as you'll see, we have more questions at the moment than answers, but we need to pay very careful attention to the emergence of this new variant. So let's start off by reminding ourselves what a virus variant is, and remembering that all viruses change over time. And in particular, they undergo changes in their genetic code, and these changes may or may not affect their biological properties. We'll look at some of these in a moment. Those viruses which undergo these changes, which we also call mutations, and which have changes or mutations that were not found in the original ancestor virus, we call variants. So over time, we expect to see variants of viruses emerging. We're familiar with this with other infections, the classic one being influenza every year. In the same way, SARS-CoV-2 has the ability to change and has changed. This is a natural phenomenon, but we have a little concern that if you have a community such as ours, where there is ongoing transmission of the virus, which has been the case since the first appearance of this infection, and where the community is partially immune because of low vaccination levels, that you might end up in a situation where the partial immunity of a community encourages the virus to undergo changes in order to escape that incomplete immunity. And that may well be a factor that operates in the emergence of all of the viral variants that we see in the COVID setting. So when a variant arises, it's designated by several terms. It may be called a variant of interest. That is where the mutations that have been recognized change some feature of the virus, but the change that occurs is thought to be of minor clinical or epidemiological importance. We then have variants of concern, where the changes that occur are significant in terms of the disease itself and its spread within a community, or even with regard to the treatment that's applied. So we worry about possible changes in the ability for the virus to spread from person to person, or the ability of the virus to evade immune control, or to evade any of the interventions that we use. And finally, of course, we want to be sure that if there is a variant, we're able to detect it in the laboratory. And I'll show you some interesting data on this in a moment. And lastly, there are variants of high consequence, which fortunately we have not yet encountered and hope never to encounter, because these are viruses which have such extensive mutations and such extreme behavioral changes that it's almost like starting the epidemic all over again. So the story of Omicron really goes back about 2 weeks. And in our virology laboratory at Lancet, we started to notice an unusual pattern of PCR results for COVID. So this slide shows you what a typical PCR reaction result looks like, and I'll quickly walk you through it and highlight what struck us. So a PCR reaction is one in which one identifies a specific target In the case of COVID, it's a gene target or several gene targets. And then one amplifies those targets and detects them. And by applying very stringent criteria to the result of the test, one can decide whether that test is positive or not. So just to quickly walk you through this here, I hope you can see the arrow, which is tracing this blue horizontal line. That is what we call a threshold of detection. And these various colored lines that you see over here are the amplification results and detection results of certain targets. So all PCR tests have a control in them. In this case, it is shown by this black line here. And we know from the shape of the curve and the way it's behaving that this is a true amplification. That control is present. And from the pattern that we see and the figures that are attached to it, we can tell that this test has worked. In this particular PCR test, we are looking for 3 specific genes that are found only in SARS-CoV-2. The first, shown by this green line, is the N gene, which you see rises above the threshold of detection very nicely and is clearly present. We are looking at a gene here called the ORF gene, which does the same thing. But curiously, the gene called the S gene is really doing nothing, and it is not being amplified, and it is not being detected at all. So this is really unusual. And we started to notice amongst the tests, the samples that were referred to us about 2 weeks ago, that this pattern was becoming more and more common, which we describe as S gene dropout. Once we started to see it happening very frequently, we were concerned that we were observing a change in the virus, that something had occurred to affect the S gene structure of the COVID virus that is circulating in our community. And as a result, our PCR was not detecting it. There clearly is virus present here. Amplifying and detecting the genes, but for some reason there is S gene dropout. And that was the first hint to us that a mutation or series of mutations might have occurred, which we had not seen before. Why do we worry about the spike gene? Well, the spike gene is shown here by this little red structure on the surface of the virus. You can see it over here. And one region of that spike, which we call the receptor binding domain, is the portion of that molecule that engages the ACE2 receptor, the ACE2 receptor being the target on the host cell, on the human cell, in the lung, intestine, kidneys, etc. And by engaging with that receptor, the virus is able to initiate infection. So anything that affects the spike protein behavior potentially can influence the virus-host interaction. And since the vaccines we are currently using all work by making antibodies to block this interaction and thereby reduce infection, could be compromised if there was some sort of mutation in the spike protein. And as you all know by now, this has been identified as something novel, this mutation, which we had hints of 2 weeks ago. And fortunately, the terminology we use now is quite simple. Thanks to the World Health Organization, we call these variants by Greek letters, and this particular one is called Omicron. So Omicron is the new circulating variant of SARS-CoV-2, which we recently identified in the Molecular and Virology Laboratory at Lancet, and that information has now been shared, as you know, worldwide. It also goes by other names depending on different classification systems. You will also hear it referred to as the B1.1.529 variant. That's another classification system called the Pango system, but we tend not to use that because it's complicated and it doesn't mean much to people outside of a laboratory. So this is what we found. When we sequenced the genes of this new variant, to our astonishment, we saw a number of mutations, and each of them is shown here by this rather indistinct text. I'm sorry about that. What was unusual about this is we'd not yet previously encountered, and nor had anybody else encountered, a virus with this many mutations in one specific region of the virus. So if I can just take you through the background of this little image, that pink line that you can see over here is one part of the spike gene. Here's the rest of the spike gene. And as you can see throughout this genetic sequence, there are multiple mutations at different sites. We also saw that there were mutations at more distant sites from the spike. And this is the reason that we were no longer able to amplify the S gene using the method which happens to be in place in our lab. In the minute we see this kind of extensive mutation, we worry about three things. Is this virus potentially more transmissible? That if it is, it could make it more contagious. We've already seen how transmissible Delta has been, and the big question is this going to be the same, somewhat more contagious, or possibly less? We can understand that or get some clues by looking at individual mutations, which I'll go through in a moment. Is there perhaps a chance that some of these mutations could enhance virulence? After all, these changes are in the region of the virus which has the primary interaction with host cells in the human. And if that is affected, could that mean that the type of illness that emerges is somewhat different? And equally important, could all these changes, which are happening in the region where vaccines are protective, could these changes allow the virus to escape from the protection of vaccines, which we have managed to build up so far, or even protection from natural antibodies following an infection with this virus rather than a vaccination? So we have some idea, and we have some idea based on historical data. Historical data deriving from previous variants that we've encountered and understanding from those what behaviors are changed when certain mutations are present. So we know that the more mutations SARS-CoV-2 has, the higher the chance that one may not be able to neutralize it. In other words, we know there is some fall off in the degree of protection that one gets with antibodies. However, please remember that immunity to COVID is a combination of antibody-based and cell-based, so that a change in the ability of antibodies to neutralize the virus may not have clinical impact because cellular immunity will still remain robust. We know that mutations around this region here allow the virus to enter the cell more easily, I beg your pardon, and transmit more easily. We saw that with Delta. This continues to be present in this variant. We know that there are some mutations here which make this virus more resistant to the action of endogenous interferon. And lastly, there are two mutations here that are seen in a distant part of the virus, what is called the nucleocapsid on this side here, which previously have also been associated with increased infectivity. So from information we've gleaned from seeing certain mutations in previous variants, the so-called alpha, gamma, lambda, and delta variants that have arisen, we have concerns that some of these behaviors may be present in the new variant, but as yet it's speculative, it needs to be determined, and those experiments are currently underway in many laboratories across the world. So we cannot say for sure that any of these behavioral changes will emerge and to what degree. What is of concern is looking at how quickly Omicron is taking over the picture in the areas where we have found it in South Africa. So this graph here just shows you what we found from genotyping the virus over the course of the epidemic. Initially, the bulk of the infections last year were attributable to what we call the beta variant. Okay, that was the so-called South African variant. There was a little bit of alpha here, the UK variant, which popped its head up, but almost in a matter of weeks, everything was eclipsed by Delta, which has persisted worldwide as the dominant variant up until now. You may remember in the local news there was a little bit of a flutter about something called the C1.2 variant, shown here in yellow. We saw a few cases of that. We never thought it would become important, and it didn't. But look what's happened in the last 2 weeks. Almost all of the genomes that we're sequencing are now Omicron. So it appears as if Omicron can outpace or outperform Delta and has rapidly taken over what is circulating in this country. And that is strong suggestive evidence that this is going to be a very transmissible virus, perhaps along the lines of Delta, or if you look at this very, very steep and rapid replacement of Delta, perhaps even a little bit more transmissible. So I'm going to finish off showing you the current data that we have. And it's not comprehensive for the entire country because I can only give you data for those regions in which our labs have specifically looked for this genome. Here's Gauteng. And what you see on the top line here is the number of tests we've performed in that week in this region. This is the percentage positivity for COVID. So as you'll see in the early part of November, positivity in Gauteng, like the rest of the country, was extremely low. And I think this is when everyone got a little bit complacent and thought we were out of the woods. But from midway through November, as you can see, the percentage positivity has started to dramatically increase. Okay. But more importantly, if we look at what's happening from an absence of S gene dropout, in other words, no variant, About a week ago, 70% of all the positives in Gauteng were S gene dropout or variant virus. And as we sit today, 96% of all the virus we're seeing in Gauteng appears to be this variant. And the same is happening in, or has happened in the Northwest. As you can see here, no hint of a variant until middle of November, when suddenly not only have numbers started to go up, percentage positivity, but at the moment, all of the positives in the Northwest are Omicron variant. In Mpumalanga, exactly the same pattern being seen. And in Limpopo, all of the infections at the moment, 100% are Omicron. So we're certainly at the beginning of something new and concerning. Does it mean we have to change anything we do? At the moment, there's no indication that different interventions are required. Are we looking at something which could be extremely serious? We don't know. What would be immensely helpful is if good clinical information could be added to all request forms for COVID testing from now on. We want to know, are these infections happening in people who have been vaccinated or not unvaccinated? If they've been vaccinated, which vaccine have they received? What sort of clinical picture are we seeing? And of course, it's going to take 2 to 3 weeks before we have any idea of whether the illness that is caused by this variant is going to be anything different to what we've seen before. So in the meantime, I know this sounds a little silly when we're facing something new and at the moment uncertain, but we do know that whatever the variant is that's circulating, it cannot cause infection unless one allows it to be transmitted. So the good old standbys of maintaining physical distance, wearing a mask and hand sanitizing, and above all, vaccinating people still hold true. And I think if there was ever a time for people to re-evaluate their commitment to vaccination, it's now. It would be wonderful if we could get all the lazy and hesitant people to go and get their vaccines, because any protection we get against COVID, whatever the variant, is going to help to markedly decrease transmission and worldwide turn this from a pandemic to an endemic state. So I'll stop talking there. And if there are a few questions or issues that need clarification, I'm happy to do that. Thank you.